But there was a major obstacle to development of mRNA-based clinical applications. When in vitro produced mRNA was delivered into cells, it resulted in low protein amounts, and additionally, it triggered an inflammatory cytokine response. This year's Nobel laureates, Katalin Karikó and Drew Weissman, started working together at the University of Pennsylvania, bringing with them highly complementary expertise. Karikó had a background in RNA biochemistry and a strong vision of RNA-based therapeutics. Weissman had a background in immunology, working with dendritic cells and their role in immune surveillance and vaccination. It was clear to them that in vitro produced mRNA must somehow differ from the mRNA made in human cells. Delivering in vitro produced mRNA into dendritic cells induced the inflammatory cytokine response, whereas the delivery of human mRNA did not. Using bacterial mRNA, shown on the right side, did also trigger the inflammatory response. Having explored several potential differences in the mRNA, they became interested in nucleoside base modifications. RNA contains 4 bases, often abbreviated as A, U, G, and C. However, these bases can get further chemically modified within our cells. It was becoming appreciated that base modifications were more prevalent in human RNA compared to RNA in bacteria. Could the lack of base modifications in mRNA produced in vitro be the critical factor that leads to inflammatory response? In a series of carefully constructed experiments, Karikó and Weissman used the T7 in vitro system to produce mRNA with various nucleoside base modifications. Each type of produced mRNA separately introduced into dendritic cells while monitoring the TNF-alpha levels, an important inflammatory cytokine. They discovered that mRNA produced with specific base modifications could almost completely abolish TNF-alpha secretion. In particular, modifications to the uridines, highlighted on the right side in orange, made a large difference. One of the most effective modified bases was pseudouridine, which is a naturally occurring uridine modification. Whereas in vitro produced mRNA triggered toll-like receptor activation, leading to inflammatory cytokines, mRNA made with pseudouridine did not. So Karikó and Weissman published their breakthrough discovery in 2005, 15 years before the COVID-19 pandemic. Realizing the importance of their discovery, they next demonstrated that base-modified mRNA resulted in higher protein expression in target cells, both in cell culture and in animals. More protein was made from each delivered mRNA molecule, and the laureates carefully dissected the cellular pathways involved. Karikó and Weissman made fundamental discoveries of the importance of base modifications in mRNA, which eliminated a major obstacle to mRNA-based clinical applications.